Okay, so for CC2 or Clinical Chemistry 2, our first topic will be about enzymes. Okay, so ano ba ang enzymes? Saan siya ginagamit or bakit meron tayong enzymes? So tandaan muna, para sa enzymes, ang ating general form or enzyme reaction is enzyme plus substrate. So interchangeable or pwede siyang magbalik. Enzyme substrate complex. Then ang kanyang final product is product plus enzyme. So enzyme ay hindi na-consume. Okay. So biological proteins that catalyze biochemical reactions. So tumutulong siya sa mga biochemical reactions natin. Either kailangan siya or minsan nagpapabilis siya ng ating reactions. So enzymes are not consumed or changed in composition. So they are found in all body tissues. So meron tayong specific enzymes para sa ating specific tissues na pwede natin gamitin either pang detect or pang measure natin ng activity. Next is enzyme is increased in serum after said injury. So halimbawa, nagkaroon ng injury na bugbog or heart attack or any other injury sa ating mga tissues, pwedeng gamitin ng enzyme pang measure ng degree or kung gaano kalala yung naging injury. Okay, so functions of enzymes. So they hydrations of carbon dioxide through respiration. So they serve as nerve induction, muscle contraction, nutrient degradation, growth and reproduction, and energy storage and use. So usually ang focus natin pagdating dito sa clinical chemistry is yung damage sa tissues. Yung ating malimit na ginagamitan ng enzymes or doon ginagamit ang enzymes sa laboratory. Okay, general properties of enzymes. So components of enzymes. So an enzyme has an active site. So may iba pa tayong mga parts niyan. So start natin sa active site. So ito yung cavity of an enzyme where the substrate binds and undergo chemical reaction. So kumbaga dito siya nagsushoot. So tandaan, ang ating enzymes ay may lock and key. So kung saan siya kasya, doon lang siya pwede. So ito, color yellow, ang ating enzyme. Itong part na to, ang ating active site. Then, ito ang ating substrate. So substrates, yung ating mga substances kung saan kailangan enzyme para ma-convert siya into another product. So once mag-fit yung ating substrate sa active site, magpo-form siya ng enzyme substrate complex. So ang ating enzyme substrate complex ay reversible. So pwede siyang bumalik sa enzyme tapos substrate lang. So kumbaga yung arrow nito, yung baliktadan, ang tawag natin parang reversible. So enzyme plus substrate nag-fit sa active site, enzyme substrate complex. Next, ang kasunod na nito is yung product and enzyme ulit. So yung substrate natin nako-convert papunta sa product, pero yung ating enzyme still retain siya as enzyme pa din. So next is the allosteric site. So allosteric site is a cavity other than the active site that binds the regulatory molecule or effector. So ang ating enzyme, di ba, kung saan siya kasya or dun sa active site, dun siya pupunta ng ating substrate. Ngayon, yung allosteric site is another part o kumbaga isa pang slot na kung saan pag may pumuntang inhibitor or ibang substance, mag-i-stop yung ating biochemical reaction at hindi magpo-proceed yung ating substrate papunta sa product. So either stuck siya doon or mawawala lang siya. Pagbalik doon, kakalas, balik siya sa kanyang substrate. So hindi siya na-convert. Gawa nitong ating inhibitor. So more on that sa ating next part nito. So yun muna, tandaan. So active site, then allosteric site. So other cavity sa ating enzyme. So isoenzyme. So isoenzymes are enzymes of similar enzymatic activity but differs in physical, biochemical, and immunologic characteristics. So usually may naglilipat lang from dextro, nagiging left, or yung isang carbon natin na papupunta sa kabila. So kung kayo nagbuo ng clay models ng ating mga compounds or chemicals, doon yun. So halimbawa, may carbon sa left side ng ating chain, naglipat siya sa right. So yun, magiging siyang isoenzymes. So apoenzymes naman is the protein portion of the enzyme subject to denaturation in which it loses activity. So siya isang portion ng enzyme. Haloenzyme, so activate enzymes or substrate formed by combination of coenzyme and an apoenzyme. So coenzyme plus apoenzyme equals haloenzyme. Last, zymogen or proenzyme, so inactivates enzyme precursors. So siya yung mga coagulation factor and digestive enzymes. So tandaan ang meaning ng mga ito. Okay. So ito yung sinasabi kong formula kanina. So terms associated with enzymes. Okay. Substrate. So binabanggit ko kanina. Ano ba tong substrate na ito? So substrate is a substance acted upon by enzymes specific for each of their particulate enzymes. Okay. So yung substance daw o isang bagay, molecule, kung saan kailangan ng enzyme at specific yung enzyme na yun para sa substrate para magkaroon siya ng chemical reaction. Okay, so enzyme natin plus substrate. So substrate ito. So reversible reaction, nagkakaroon siya ng enzyme substrate complex. Then, kapag nagtuloy or walang naging inhibitor, magiging siyang product plus enzyme. So product natin is from the substrate, na-convert siya into product. Yung enzyme, hindi naman siya magbabago into composition or sa ibang composition. So, remain enzyme siya. Itong enzyme na to, maghahanap naman ng kasunod na substrate para magkaroon ulit ng enzyme substrate complex at magkakaroon ulit ng product. So, ganun lang yan. Paikot-ikot lang yung cycle. Yan. Cofactor. So, cofactor is a non-protein substance added in enzyme substrate complex to manifest the enzyme activity. Kumbaga, yung enzyme substrate complex natin may kulang pa. So, kailangan pa si cofactor para mabuo yung ating biochemical reaction at magpatuloy dito sa ating formula. So, enzyme substrate papasok plus cofactor para magpunta dito sa ating product at enzyme. So, either kasama siya sa pag-convert or pwedeng yung cofactor natin balik ulit sa pool parang enzyme. So, basta lagi nako-convert yung ating substrate. So, two types of cofactors. Pwede siyang cofactor or prosthetic group and an activator. So, anong pinagkaiba nila? So, kapag coenzyme, siya ay organic cofactor and nucleotide. So, NAD, NADP, and vitamins. Kapag activator naman, siya ay mga metals at inorganic cofactors. Next. So, enzyme classifications. Okay, so tatandaan ito kasi usually, yung ating classification dun naka-base yung kanya or nakapangalan yung ating reaction na nangyayari. Okay, so oxidoreductase. So, catalyzes redox. So, reduction, oxidation. So, oxidoreductase. Reactions between two substrates. So, yung ating clinically significant na oxidoreductase enzymes is lactate dehydrogenase and glucose 6-phosphate dehydrogenase. So, usually, may dehydrogenase siya sa pangalan. So, oxidoreductase. Next, transferase. So, catalyzes the transfer of a group other than hydrogen ion. So, other than hydrogen na inilipat sa ating chemical reaction, transferases ang ating yan. So, ALT, AST, GGT, and CK. So, ano mga meaning nito? So, abangan nyo sa ating discussion ng ating enzymes. So, hydrolase. So, hydrolase naman catalyzes hydrolysis of bonds. So, hydrolysis is the splitting of substrate involving water. So, hydro. So, example niya is esterase. Ating esterase ay ALP, ACP, cholinesterase lipase. Ang esterase ay splits esters into acid and an alcohol. Peptidase. So, trypsin, pepsin, leucine, aminopeptidase. So, atin namang peptidase breaks down peptides into amino acids. Last, glycosidase. So, amylase and galactosidase. So, galactosidase catalyzes the hydrolysis of glycosidic bonds in complex sugars. So, breakdown of sugars. Itong isa, amino acids. Yung isa, esters. So, esterase, peptidase. So, parang peptide, di ba? Glyco. So, glyco meaning glucose or sugar. Peptidase. So, peptide chains natin. Ester, esterase. Liases. So, catalyzes removal of substance without hydrolysis. If hindi hydrolysis, yung naging pagtanggal sa ating substances, liases yon. So, aldolase, pyruvate decarboxylase, glutamate decarboxylase, tryptophan, and tryptophan decarboxylase. Next, isomerase. So, isomers naman ulit. So, isomer catalyzes interconversion of geometrical, optical, or position isomer. So, nalilipat lang yung ating parts. So, nalipat lang ulit. Parang, yung carbon from the left, pwede malipat sa carbon from the right ng same carbon na kinakabitan niya. Ligases, so joining naman. So, pag ang ating nangyaring biochemical reaction is joining, so ating enzyme na yun ay ligase. So, glutathione synthase. General properties of enzymes. So, enzyme kinetics. So, ito yung kung gaano kabilis yung ating reaction ng mga enzymes. Okay. So, una is the substrate concentration. So, substrate concentration is called first order kinetics. So, tandaan, first, F-I-R-S-T, substrate. First, substrate. So, first order kinetics is substrate concentration. So, the reaction rate is directly proportional to the substrate concentration. So, yung speed ng ating biochemical reactions ay nakadepende kung gaano kadami yung ating substrate. Kasi, sobra yung enzyme dito. So, madaming enzymes na available kaso kulang yung substrate. So, kailangan ng mga substrate na pupuntahan ng enzymes para magkaroon ng conversion. So, lack or kulang ang ating substrate sa first order kinetics. First, FIRS, substrate concentration. Next, enzyme concentration. So, enzyme, 0. N0. Zero order kinetics. So, kapag zero order kinetics, enzyme concentration naman. So, reaction rates depends on enzyme concentration and there is a substrate excess. So, madami yung substrate, kulang naman yung enzyme. So, nag-aantay yung substrate para sa mga enzymes para magpatuloy sa ating biochemical reaction. So, para saan ba itong dalawang to? So, these two are used for the measurement. So, kung paano or ano naging problema sa ating measurement ng ating enzyme and substrates. Next, pH. So, most physiological reactions occur in the pH range of 7 to 8. So, they are controlled by a buffer. So, buffer is a substance that is added to control the pH. Temperature. So, increasing temperature equals increased rate of chemical reactions. So, paano natin masasabi yun? For every 10 degrees of Celsius increase sa ating temperature, the reaction will be doubled. Okay, so halimbawa 37. Gusto natin bumilis yung reaction. Dinagdagan natin ng heat. So 37 plus 10, 47. Yung speed is na doble na. So enzyme is active at 25, 30, and 37 degree of Celsius. So kapag 40 to 50 degrees of Celsius na, significant denaturation of enzyme. So kapag nandito ka sa range na to, 40 to 50, masisira or mawawala na ng bisa yung ating enzymes. Cofactor. So, activator, metallic, non-metallic coenzyme, and secondary substrate. Okay, so dito tayo sa inhibitor. So, ano ba yung inhibitor? So, inhibitors are substances that prevent or stop the biochemical reactions of our enzyme and substrate. So, competitive inhibitor, so meron tayong tatlo nito. Una, competitive inhibitor, so palaban. So, kapag competitive inhibitor, they bind to active sites. So, ano yung active site natin kanina? Ano yung nakakabit doon? Okay. So, ang nakakabit sa ating active site is yung substrate. So, ibig sabihin, ang competitive inhibitor natin nakikipag-agawan sa pwesto ng substrate. So, active site. Next, non-competitive inhibitor. So, non-competitive inhibitor does not bind to the active site but to the allosteric site. So, sa kabilang part siya na kikikabit yung ating non-competitive. Last, uncompetitive inhibitor binds the enzyme substrate complex. So, sa kabuuan ng ating enzyme substrate, siya nagbabind. So, ano bang itsura nitong tatlong ito? Okay. So, competitive inhibitor. So, ito yung ating regular na enzyme substrate complex and reaction. So, yung color gray is our enzyme and our substrate is yellow. So, kakabit siya sa active site. Tawag dito, enzyme substrate complex. So, yung ating substrate na-convert na into product. Okay, gets? So, paano pumasok yung competitive inhibitor? Dito siya. So, enzyme ulit natin, itong color gray. So, yung ating inhibitor, itong color blue. So, sa active site siya kakabit. Tapos, nakakabit pa din siya sa active site. Nakakabit sa active site, hindi makapunta ang ating substrate. So, hindi natuloy ang ating reaction. Stop doon. So, hindi magkakaroon ng product since nakastop or hindi makapunta si enzyme dahil sa ating inhibitor. So, competitive, dun siya mismo nakakabit sa active site. Next, non-competitive inhibitor. So, saan siya? Does not bind to the active site but to the allosteric site. Okay. So, dito sa left part, yung normal reaction ng ating enzyme. Right side, sorry. So, right side, yung normal reaction natin ng enzymes. So, color gray is the enzyme. So, papasok ang ating substrate. So, color yellow. So, shoot siya dito, diba? Ayun. So, kasyang-kasya siya dito. So, yung ating enzyme and substrate complex is formed. So, magtutuloy siya sa reaction and magkakaroon na siya ng product. So, ayun yung products natin. So, maalin dito sa dalawang to or pwedeng both itong dalawa or pwedeng byproduct yung isa. Basta, product na to. So, nagtuloy yung ating reaction dito sa kanan. Sa kaliwa, pumasok si inhibitor sa kabilang butas. Kaya, nung pumunta naman si enzyme, ayun yung enzyme, mag-i-stop na siya dyan, hindi magtutuloy yung reaction. So, patay na ulit yung ating reactions na yun. So, no product will be formed due to the non-competitive inhibitor. So, hindi siya nakipag-agawan, nakishare siya doon. So, ito yung una natin, nakipag-agawan siya doon sa slot. So, hindi pwedeng matuloy ang ating reaction. So, this is reversible. So, pwedeng umalis, pwedeng hindi. Okay. So, ito nga. So, nandun, nakishare siya. So, hindi natuloy ang reaction ng ating substrate. Last, uncompetitive. So, dito, nagbabind siya sa lahat naman. Okay. So, enzyme, nakapunta yung ating substrate, pero magbablock or makikibind si uncompetitive at hindi natutuloy yung reaction. So, stop na din siya dito. So, measurement of enzyme activity. So, enzyme quantitation is the measurement of catalytic enzymes. So, activity is related to the concentration. So, usually, photometric. So, kapag anong tinutukoy nito? Activity is related to the concentration. So, kapag mataas yung concentration, mas mabilis yung ating activity or mas intense. So, sa intensity kasi photometric. So, increase in product concentration, decrease in concentration, and increase in concentration of altered enzymes. So, yan yung mga pwedeng maging reactions. Pero photometric, so sa intensity ng color, yung ating mine-measure. So, meron tayong dalawang type ng measurement, either fixed or continuous. So, mas maganda kapag fixed kasi hindi na magbabago. Mas madali yung computation para dito compared sa continuous time. So, kapag fixed, may endpoint or may dulo. So, reactions are combined and the amount of reaction is measured. So, follow the procedure. Then, after the incubation, kung meron, kapag sinabing 15 minutes, measure at 15 minutes, tapos compute ka na. Kapag continuous time naman, kinetic siya. Measures at specific time intervals, then continuous yung measurement niya. So, minsan dalawa, minsan tatlo yung measurement na ginagawa. So, kapag continuous or kinetic, follow procedure. Then, after incubation, kung meron, sasabihin niya, measure the concentration. So, babasahin mo siya yung absorbance niya. So, feed mo sa machine. Then, sasabihin nun, or after 1 minute, feed mo ulit. After 1 minute, feed mo ulit. So, ilang measurement tayo doon? Tatlo. So, 3 beses yung measurement, mas mahaba-haba yung computation compared sa ating fixed time. Okay. So, dito muna matatapos ang ating enzymes. And the next part will be about specific enzymes na. Sunod-sunod kung ano na yung mga clinically significant enzymes natin na meron sa chemistry. Thank you.